That is Martin Serrwanda from Uganda. He's a research scientist, a PhD. He's doing his PhD at the Artemia Reference Center. He's also a lecturer at the School of Agriculture and Environmental Sciences at the Mountain Moon University, with interests in embryology, laviculture, live feed, and aquaculture, smart aquaculture. So Martin, please go ahead with your presentation.
Thank you, Betty, and all the organizers for this webinar. And I'm here to give you the story of Artemia activities in Uganda. As we all know, Uganda is a landlocked country located on the eastern side of Africa, but we share part of the Rift Valley. That's the western part of the Rift Valley. And on the floor of the western Rift Valley, we have several saline crater lakes that could be possible habitats for Artemia. So that's a picture of Lake Katwe where we do local salt mining, and it's the same place that could be used for Artemia production in this country. Next slide, please. Broadly, our saline lakes have been categorized into three classes. We have the carbonate chloride lakes, where the main anion is a carbonate or a bicarbonate with a chloride less than 50%. But the important cation is mainly sodium, and these are basically Lake Chikorongo and the Nyamnuka. We have sulfate chloride lakes, where the dominant anion is carbonate, and still the dominant cation is sodium, and these are majorly Mahiga, Maseche, and Bagusa. Chloride lakes, which are of major interest for Artemia production, have more than 50% chloride as the dominant anion, and still the important cation is sodium. This is where we have Lake Katwe, Bunyampaka, and Kitagata. Most of my talk further will be on Lake Bunyampaka. We also started with a survey to see whether we have natural occurrence of Artemia in these saline lakes. And this work was produced by a colleague in 2015, and there was no Artemia in our saline lakes. So we picked water from lakes Katwe, Maseche, and Bunyampaka and tried a laboratory culture for Artemia. We hatched Artemia and cultured this Artemia to reproductive stage. The only twitch that we did, we had to filter the water because it was too viscous. It is just like an algal soup. So it is too thick for the Artemia to filter. So we aided the process by filtering it and producing a clear water that we used to culture. And this work was done at the ARC in Belgium. Next slide, please. So we cultured Artemia for 2 weeks initially until we saw the riding couples, and we were comparing the Great Salt Lake Artemia franciscana and the Vinchao strain of Artemia franciscana. And in this, we saw that the Vinchao strain was performing much better than the GSL at that level. Why we chose those two strains? The GSL is the most dominant strain on the market, while the Vinchao strain is the most inoculated strain worldwide. Next slide, please. Next slide. So when we looked at the reproductive parameters, we saw that this was an experiment that was carried out at a salinity of 30, 50, and then the control was seawater at 35 grams per liter. The most interesting results were at age at first offspring, when we saw the first offspring from what we had cultured, realized that our Artemia cultured at the 50 grams per liter salinity was producing later. It took a lot of time to produce its first offsprings than the control group, and then the control group was really comparable with the 30 grams per liter culture. If you look at the offsprings per female per day, The one at 50 grams per liter salinity produced less offspring per female per day than the rest. And then if you looked at the total offspring production from those cultures, still the one at 50 produced much less than that of the control, while the one of 30 really performed well. Next slide, please. So basically in Uganda, we haven't yet inoculated in our wild lakes, but we've done the preliminary laboratory cultures. So we want to go to the next stage of performing bucket and tank trials. Then after that, we shall do field pond trials. We also intend to look at the algal diversity within these lakes so that we could look for what we can feed onto the Artemia naturally in order to promote Artemia production in this country that is looking so much ahead in terms of aquaculture production. Thank you for listening to me.